Welcome back to the Deep Dive. Today, we are strapping on the virtual reality headset and shrinking down to the nanoscale to conduct really an exhaustive tour of the cellular structures responsible for life's most essential functions: synthesis, quality control, and transport. We are diving deep into the biosynthetic machinery, this interconnected world of the endoplasmic reticulum and the Golgi apparatus.
That's right.
Our mission really is simple: to stop treating these as just static diagrams in a textbook and truly understand the dynamic molecular choreography that lets a cell synthesize, fold, modify, and ship almost every single protein and lipid it needs. It needs to grow, maintain itself, communicate. I mean, this isn't just basic cell biology. This massive infrastructure is running a high-stakes 247 manufacturing operation. It's constantly renewing every structural protein, every enzyme, every phospholipid membrane the cell requires.
It is an absolute incredible feat of logistics, yeah. And what enables this staggering level of productivity and precision is that fundamental principle of life: compartmentalization. For you listening, understanding the necessity of these membrane-bound organelles is really key.
Why is that isolation so essential, though? I mean, if the cell wants to make a new lipid, why can't the enzymes just float freely in the cytosol? What's the big deal?
Well, it really boils down to both safety and efficiency, and these two concepts are, inextricably linked. On the safety front, many cellular processes actually require pretty harsh conditions. If you're going to degrade waste or, say, misfolded proteins, you need highly acidic environments or powerful degradative enzymes. Those would otherwise injure the cell's own components if they were just loose. So compartmentalization keeps these potentially toxic metabolites or enzymes contained.
Okay, damage control. That makes sense.
And then on the efficiency side, isolation allows the cell to create unique microenvironments. You can maintain a very specific, really high concentration of calcium ions, for example, within the ER, or a unique pH and redox state that's necessary for specific enzymatic reactions, like forming disulfide bonds. These are like specialized chemical vats, each with its own perfectly calibrated internal climate, optimized for just one step in a complex pathway.
Got it. Specialized zones for specialized jobs. And when we talk about synthesis, the endoplasmic reticulum, the ER, that's the foundational site, right? This is where it all kicks off.
Absolutely. The ER is the central hub. It's responsible for synthesizing every single transmembrane protein and all the lipids needed for the plasma membrane, the cellular organelles, and even the ER itself. Plus, it handles the initial synthesis and modification of proteins that are destined to be secreted outside the cell.
And its structure is unique. It's not just a bunch of distinct sacs floating around.
That's right. Physically, the ER is this stunning labyrinth. It's a single, massive, continuous structure. It's composed of a mesh-like, interconnected maze of branching tubes and these flattened sacs called lamellae. Crucially, they all enclose a single continuous internal space, which we call the ER lumen.
And that lumen leads us to one of the most, well, conceptually fascinating facts about the cell. The inside of the ER is, topologically speaking, the outside world.
Precisely. If you sort of imagine the ER membrane is just an extension of the plasma membrane folded inwards, then the ER lumen is equivalent to the extracellular space. Everything that enters the ER is functionally outside the cytoplasm. And this structural relationship has profound implications for how the cell synthesizes and modifies its products, especially membrane proteins. Like you said, the cell has to be obsessively precise about which side of a protein faces the cytosol and which side faces the lumen, or ultimately the outside environment.
Right. And to ground this massive system in reality, our sources cite hepatocyte data, a typical liver cell. In that cell, the rough ER alone occupies about 9% of the total cellular volume.
9%, and that's just the rough part. The Smooth ER and the Golgi apparatus combined account for another 6%. So we're dedicating, 15% of the entire cell volume just to this complex, highly active biosynthetic and processing infrastructure. It really underscores the cell's primary need: constant renewal and synthesis.
Okay, let's start the assembly line then, at the rough ER, or RER. It gets its name because its cytosolic face is heavily studded with ribosomes, the protein synthesizing machines, giving it that characteristic rough look under an electron microscope.
Exactly. And this is where the cell commits to the destiny of a protein. The critical sorting decision happens right here. Proteins intended for the plasma membrane or secretion or other organelles like the lysosome are assembled on these RER-bound ribosomes. In contrast, proteins intended to function inside the cytoplasm, like, say, structural proteins or metabolic enzymes, those are synthesized on free ribosomes just floating in the cytosol.
Okay, so the core question here is the trigger. How does the cell know to direct a specific ribosome complex to the RER membrane versus letting it finish translation freely? It must require some kind of instantaneous recognition mechanism.
It does. It relies on a very specific kind of zip code called the signal sequence. As translation begins on a free ribosome, if the nascent polypeptide chain, the growing protein, possesses this specific sequence, usually a stretch of hydrophobic amino acids near its N-terminus, it triggers a whole cascade.
Okay, a cascade. And what does that cascade look like at the molecular level? How does it physically get there?
Well, as soon as that signal sequence emerges from the ribosome, it's immediately recognized and bound by a cytosolic chaperone complex. It's called the signal recognition particle, or SRP. The SRP binding actually pauses translation temporarily, and then it physically guides the entire complex, the ribosome, the mRNA, and the partially synthesized protein over to the ER membrane.
So the SRP is like the ferry, bringing the machinery to the correct dock. But what's the dock itself made of?
The dock is a specialized protein channel embedded right there in the ER membrane. It's called the Sec61 translocon. The SRP complex docks onto its specific receptor on the ER membrane, which is right next to or associated with the Sec61 channel. Once it's docked, the translation pause is released, the nascent polypeptide chain is threaded through the Sec61 channel, and translation resumes. This is what we call co-translational translocation.
Co-translational translocation, meaning it's happening at the same time.
Exactly. The protein is inserted into or across the ER membrane as it is being translated, not afterwards. It's incredibly efficient.
That is highly efficient. So once the protein is threaded through, what happens to that signal sequence zip code that started the whole process? Seems like you wouldn't want to leave it attached.
You're right. It's promptly removed. As the signal sequence enters the ER lumen, an enzyme called signal peptidase recognizes the sequence and just cleaves it off. SNP. The protein then begins its post-translational life inside the ER lumen. If it's a secreted protein, it's released entirely into the lumen. If it's a transmembrane protein, specific hydrophobic stop transfer or start transfer sequences within the protein anchor it within the Sec61 channel. The channel then opens laterally, kind of like a clamshell, to integrate the protein right into the lipid bilayer.
Okay, so once a protein is inside the RER lumen or embedded in the membrane, we enter this really critical phase, folding and modification. This is the first essential quality control checkpoint, right?
Indeed. The RER lumen has this unique chemical environment that's optimized for these modifications. It's highly oxidative, for instance, which is necessary for forming disulfide bonds. These covalent links are essential for stabilizing the tertiary and quaternary structure of many secreted and surface proteins, and they just can't form correctly in the highly reducing environment of the cytosol.
Ah, another reason for compartmentalization. And this is where the chaperones really take center stage, making sure the protein assumes its correct 3D shape or conformation.
Yes. The primary chaperone responsible for checking protein folding inside the ER is BIP, that's BIP, or binding immunoglobulin protein. BP is an ATP-dependent chaperone. It binds to newly synthesized proteins, particularly bits that are hydrophobic or look misfolded. It uses cycles of ATP hydrolysis using energy to repeatedly try and pull the protein into the correct fold.
I remember reading that the initial sugar tags, the N-link glycosylation, they begin here too. How does that modification link into the quality control process?
Oh, it's beautifully integrated. N-linked oligosaccharides, these are sugar trees, are added to specific asparagine residues in the RER. Then, specific ER chaperones like calnexin and calreticulin act as lectins. Lectins are just proteins that bind to carbohydrates. So they specifically bind to these newly added sugar tags on the proteins, holding them in the ER, giving them time to fold correctly. If the protein fails the folding test, it actually gets reglucosylated, another sugar added, giving it another chance to bind to the lectins and try refolding.
So the protein gets multiple attempts at folding, guided by chaperones and marked by these sugar tags. It sounds quite forgiving, actually. But what happens if it ultimately fails? If you just can't get it right?
This is where the system is absolutely ruthless in its efficiency. If a protein fails to fold appropriately, or oligomerize, form its complex with other subunits, or pass the chaperone check after multiple attempts, it's condemned. It gets marked for ER-associated degradation, or ERAD. The misfolded protein is retrotranslocated, basically pulled back out of the ER lumen, across the membrane, and into the cytosol. There it gets tagged with ubiquitin and degraded by the proteasome. It absolutely does not get to exit the factory floor in a defective state.
Wow. Okay. Ruthless is right. So from the RER, where protein destiny is decided and quality is rigorously checked, we transition into the smooth ER, the SER. This is the domain that's contiguous with the RER but lacks ribosomes. You mentioned in many cells it's mainly a transport zone, but in specialized cells it's a chemical powerhouse.
It really is. It's the cell's specialized chemical workshop, focused almost entirely on lipid synthesis and the metabolism of hydrophobic molecules. Now, while the proteins that synthesize the lipids are often made in the RER, the actual lipid synthesis itself happens on the cytosolic leaflet, the side facing the cytoplasm of the SCR membrane.
Can you walk us through the basics of that lipid production? How does it build the membrane?
Yeah, phospholipid biosynthesis is a prime example. The precursor molecules, like fatty acids, are imported from the cytosol. Enzymes embedded in the SCR membrane then assemble the finished phospholipid molecules right there on the cytosolic face. But because this process only happens on one side of the membrane would quickly become unstable and asymmetrical if nothing else happened.
Right, one side would grow much faster.
Yeah.
How does the cell correct that instability?
It uses enzymes called scrambloses and flippases. Scrambloses randomly move phospholipids between the two leaflets of the bilayer to sort of equalize the surface area, ensuring stable membrane growth. Later on, as lipids move to other places like the plasma membrane, specific enzymes called flippases will actively transport particular lipids to set up the critical asymmetry needed for things like cell signaling.
Okay. And where is this SER specialization most visually obvious? Which cells have tons of SER?
You see it prominently in any cell requiring extensive steroid hormone production. So, for example, cells in the testes, ovaries, or the adrenal cortex that synthesize testosterone, estrogen, or cortisol, they have massive complex networks of SER. It's the primary site for converting cholesterol derivatives into these essential steroid hormones.
Makes sense. The other major function you mentioned earlier, especially vital in the liver, is detoxification.
Yes, detoxification is a cornerstone function, particularly in hepatocytes' liver cells. The SER is just loaded with enzymes that catabolize lipid-soluble molecules. These are often drugs, toxins, or environmental pollutants.
How does that chemical detoxification work exactly? How does it make something less toxic?
Well, the primary system involves the cytochrome P450 enzyme family. These enzymes are what we call monooxygenases. They introduce polar hydroxyl groups, that's an OH group, onto these lipid-soluble compounds. This hydroxylation process makes the hydrophobic molecule more water-soluble. And once it's water-soluble, it can be easily transported through the circulation and excreted, typically out via the urine.
And this capacity isn't fixed, right? The SER physically adapts to the chemical load the cell is experiencing.
Precisely. It's highly dynamic. If you are repeatedly exposed to certain compounds metabolized by the P450 system, for example, the drug phenobarbital, the liver cells, will dramatically increase their SER surface area and the concentration of these enzymes. This is known as SER hyperplasia. It's a rapid physical cellular adaptation to increase toxic load. It really demonstrates the immediate structural response of this biosynthetic machinery to environmental demands.
Well, interesting. So the cell literally builds more detox machinery when needed. Beyond synthesis and detox, the SER has this fundamental, critical role managing the cell's most universal signaling ion, calcium.
Absolutely. The SER serves as the main intracellular calcium storage tank, sequestering it away from the cytosol where levels are kept extremely low. The calcium concentration inside the SER is thousands, maybe tens of thousands of times higher than in the surrounding cytoplasm. This sequestration is actively managed by dedicated protein pumps, most notably the SERCA pump that stands for Endoplasmic Reticulum calcium ADPase. It uses ATP energy to pump calcium in.
So if the SERCA pump is constantly pumping calcium in, how does the cell release it when a signal arrives? It needs to be fast.
It is very fast. Extracellular signals trigger receptors on the plasma membrane, and this often leads to the production of second messengers inside the cell, like IP3 inositol trisphosphate. IP3 then diffuses through the cytosol and binds to specific channels, the IP3 receptors, which are embedded in the SER membrane. These channels open rapidly, causing a massive, immediate efflux of that sequestered calcium out into the cytosol. This sharp spike in cytosolic calcium then mediates countless processes, from immune activation to metabolism, muscle contraction, and even, under certain conditions, triggering apoptotic cell death if the signal is sustained and strong enough.
And in muscle cells, the SER is so dedicated to this calcium handling function that it gets a whole new name, right?
It does, yeah. It's called the sarcoplasmic reticulum, or SR. It is the specialized SER in muscle cells, and it's entirely dedicated to the rapid cyclic release and recapture of calcium ions. This calcium cycling is the sole mechanism regulating muscle contraction and the subsequent relaxation. The highly structured organization of the SR ensures that this calcium flux is perfectly synchronized across the entire muscle fiber, allowing for instantaneous, coordinated physical movement.
Okay, so we've successfully synthesized and folded our proteins and lipids in the RER, or specialized our lipids in the SER. Now, the completed products need further modification, specific tagging, and dispatch to their precise destinations. The product gets packaged into small transport vesicles that butt off the ER and shuttle into the Golgi apparatus.
Exactly. This is the handoff point. The Golgi apparatus is the cell's complex post-office and processing center. If you think of the RER as the manufacturing plant, the Golgi is the finishing, packaging, and sorting line.
Structurally, the Golgi is pretty distinctive, those stacked cisternae.
Yes, it consists of a stack of flattened membrane-bound sacs known as cisternae. Typically 4 to 6 stacks, though it can vary quite a bit depending on the cell type and its activity. Crucially, the Golgi maintains a defined polarity, which reflects the directional flow of modification.
Let's break down that progression. We always hear about the cis face and the trans face.
Right. The cis-Golgi network, or CGN, is the receiving face. It's located nearest the ER. Vesicles from the ER fuse here. The trans-Golgi network, TGN, is the exit face, located near the plasma membrane. This is where finished products leave in new vesicles. In between, we have the medial cisternae. And modification happens sequentially as products move through the stack, from cis to medial to trans.
Now, our sources highlight that the mechanism of movement isn't static, but dynamic, involving this concept of cisternal progression or maturation. Can you detail what that means? It sounds different from just passing cargo along.
Yeah, the older model suggested products moved via small vesicles, hopping between fixed cisternae like cargo on stationary platforms. However, the prevailing model now is the cisternal maturation model, or CMM. It suggests a much more dynamic process. Basically, new cis-cisternae are constantly forming by the fusion of those ER-derived vesicles. These cisternae then physically mature, moving forward or progressing toward the trans face, kind of like steps on an escalator. As they move, they gradually acquire the enzymes for later steps and shed the enzymes for earlier steps.
Wait, but if the cisternae themselves are moving forward, how do the specific signature enzymes that define the cis stack, say the cis-Golgi enzymes, how do they stay in place to process the next incoming batch of vesicles from the ER?
That's the elegant complexity of the system. It's a massive two-way street. While the cargo progresses forward through this cisternal maturation, the Golgi-resident enzymes themselves are constantly being recycled backward. Small coated vesicles, specifically Copi-I-coated vesicles, bud off the maturing cisternae. They capture the necessary Golgi-resident enzymes and shuttle them retrograde from trans back toward cis to maintain the correct enzyme concentration and specificity in each stack. It's a really high-traffic system that cleverly manages the forward flow of product and the simultaneous reverse flow of the processing tools.
That is clever. Okay, now let's talk about the specific modifications that really define the Golgi, this advanced glycosylation, the complex sugar coating of proteins and lipids.
This is absolutely the Golgi's primary identity. While those N-linked oligosaccharides, the ones attached to asparagine, start their journey in the ER, they are extensively processed here in the Golgi. The Golgi is where those initial sugar trees are first pruned and then elaborated into generally 2 main forms. The complex oligosaccharides, or the high mannose oligosaccharides, depending on the specific enzyme activity in the cisternae.
And the Golgi introduces an entirely new type of sugar tag as well, right?
Yes, O-linked glycosylation. This involves adding sugar moieties, often one by one, to the hydroxyl groups of serine or 309 residues on the protein. Functionally, these complex sugar adducts, both N-linked and O-linked, are far more than just decoration. They are crucial functional tags that dictate a molecule's behavior, its stability, its interactions.
How do these tags determine function in the real world? Can you give some examples?
Oh, they are absolutely vital for several roles. They influence the stability and solubility of secreted proteins. They are critical for cell or cell matrix recognition. Think of your blood type antigens, ABO. Those are actually carbohydrate tags on cell surface molecules. And they also act as clearance signals. For instance, as cells like platelets or red blood cells age, terminal sialic acid groups on their surface proteins get removed. This exposes underlying sugar structures that are recognized by receptors, mainly in the liver, leading to the clearance of these senescent or old cells from circulation.
So the sugar code tells the body when it's time to take out the trash, essentially.
Okay,
Once all the processing and modifications are complete, we arrive at the final decision point, the trans-Golgi network, or TGN. This is the shipping department where products get their final destination label.
exactly. The TGN is the most structurally dynamic region. You see lots of vesicles budding off. Here, proteins and lipids are sorted into different types of vesicles. Each vesicle gets a specific protein coat that targets it towards one of 3 primary destinations. Those are 1, the secretory pathway for export outside the cell, either constitutive, meaning constant, or regulated, meaning stored until signaled. 2, the plasma membrane for integration, or 3, another specific organelle, like the lysosome.
The sorting mechanism must be highly precise, involving specific molecular zip codes again. Can we use the classic example of how the Golgi tags a protein destined for the lysosome?
Yes, the lysosomal tagging system is the canonical example of Golgi precision. Lysosomes, as you know, are the cell's recycling centers, and they need highly specific hydrolytic enzymes called acid hydrolases to function. These enzymes are synthesized in the RER like other proteins. When they reach the cis Golgi, a crucial modification occurs. A mannose sugar on the enzyme gets phosphorylated, creating the mannose 6-phosphate tag, or M6P tag.
Okay, so it gets this M6P tag in the early Golgi. What happens once that tag is applied? How does it get sorted in the TGN?
Well, in the TGN, there are specialized transmembrane proteins called M6P receptors. These receptors specifically bind only to those M6P modified lysosomal enzymes, nothing else. The receptor ligand complex is then packaged into specific transport vesicles, often with a clathrin coat. These vesicles are specifically targeted to fuse with an intermediate compartment called the late endosome, or pre-lysosome. The environment inside this compartment is acidic, and the key is that the decrease in pH in the pre-lysosome causes a conformational change in the M6P receptor, which makes it release the lysosomal enzyme. The enzyme is now free to function inside the maturing lysosome, and the M6T receptor is recycled back to the TGN in another vesicle to pick up more cargo. This simple but elegant tagging system ensures that these dangerous hydrolytic enzymes only end up where they belong and never accidentally get secreted or sent to the cell surface.
That's a fantastic system. And just looking at cells that specialize in secretion gives us a visual cue of the Golgi's importance you mentioned.
Absolutely. The Golgi complex's size correlates directly with the cell's secretory activity. In highly specialized secretory cells, like plasma cells, which are churning out huge amounts of antibodies, or the mucus-secreting goblet cells in your gut lining, the Golgi is so large and active that it actually appears as a distinct, clear, or pale area right next to the nucleus on standard histological stains. Pathologists call it the paranuclear HOF. Seeing that pale zone tells you instantly, this cell is manufacturing and shipping a lot of product.
Right, a clear sign of high export activity. Okay, we've established the RER has this intense quality control system. But what happens when the factory gets overwhelmed? When the rate of synthesis of new proteins is just higher than the RER's ability to fold them correctly?
That critical imbalance leads to a state we call ER stress. And this triggers a highly complex and absolutely essential survival mechanism called the unfolded protein response, or UPR. This occurs when misfolded proteins start to accumulate in the ER lumen, exceeding the capacity of chaperones like BP to manage the backlog.
So the cell senses this overload. How does it initiate the response? This isn't just one reaction, right? It's a coordinated, multi-pronged signaling attack.
It really is. It's managed by a network of 3 parallel transmembrane signaling proteins that are embedded in the ER membrane itself. Under normal conditions, all 3 are kept inactive because they are bound to that main chaperone by P, but when misfolded proteins accumulate, BP preferentially binds to them, letting go of the sensor proteins. This release activates the sensors. These 3 sensors are named IRE1, PR, and ATF6.
Okay, 3 sensors. Let's start with IRE1. What does it do when it gets activated?
IRE1 is the most conserved sensor found across many species. When activated, it functions as an enzyme, specifically an endonuclease on the cytosolic side. Its primary target is an mRNA molecule known as XBP1. IRE1 literally cuts or splices this XBP1 mRNA molecule in the cytosol. This splicing event creates a new, highly active transcription factor from the XBP1 message. This activated, spliced XBP1 then translocates to the nucleus and massively upregulates the transcription of genes coding for ER components, particularly more chaperones and factors involved in the ERAD degradation pathway. It's essentially the cellular signal to build more factory capacity and hire more folding assistants and quality control inspectors.
Okay, so IR1 increases capacity. What about the second sensor, PRK? What's its role?
PRK acts more like a protective throttle. When PRK is activated, it acts as a kinase. It phosphorylates a critical translation initiation factor called EI2. Phosphorylation of EI2 broadly reduces the global rate of protein synthesis across the entire cell. This is like hitting the emergency brake. The cell dramatically slows down manufacturing of all new protein to allow the existing folding machinery time to catch up with the backlog.
Smart. Reduce the inflow while you increase the processing power. And what role does the third sensor, ATF6, play in this sophisticated response?
ATF6 acts as a regulated transcriptional activator, but in a slightly different way. When it's released from BAP, it actually travels to the Golgi apparatus. There, it's cleaved by specific proteases. The N-terminal fragment, which is the active transcription factor part, then travels to the nucleus. Much like the spliced XBP1 from the IRE1 pathway, this ATF6 fragment upregulates the expression of specific UPR target genes, again further enhancing chaperone production, and also, interestingly, lipid synthesis capacity, likely trying to expand the ER membrane itself to dilute the minfolded proteins.
Wow. It sounds like a remarkably sophisticated feedback loop designed entirely for recovery. But you hear that if the stress is too profound or goes on too long, this system can actually initiate cell death, right?
Absolutely. The UPR is fundamentally a pro-survival response, but it's definitely a double-edged sword. If the stress is chronic or just overwhelming, if the system fails to correct the overload and restore homeostasis, the signaling pathways, particularly involving IR1 and PERK, can transition from survival mode to actively signaling for apoptosis or programmed cell death. Essentially, the ER is constantly monitoring its own integrity, and if that integrity cannot be restored, the cell initiates self-destruction rather than risk continuing to produce potentially dangerous misfolded protein aggregates, which can be toxic.
That is a critical balance, and it brings us perfectly to a case study that really illustrates the power and sometimes the detrimental effects of RER quality control in human pathology, the cystic fibrosis transmembrane regulator, or CFTR protein.
Yes, this is perhaps the most illustrative example of the sheer inflexibility, you could say, of the ER's quality control system when things go slightly wrong. The most common cause of cystic fibrosis is a specific genetic defect. It's a deletion resulting in the absence of just a single amino acid phenylalanine at position 508. This mutant is often called delta F508C FTR.
That seems like an incredibly subtle defect, one missing amino acid out of almost 1,500 to cause such a severe systemic disease.
It is subtle in terms of the primary sequence, yes, but it's catastrophic in terms of the final conformation or shape of the protein. The absence of that one amino acid causes the CFTR protein to slightly misfold as it's being synthesized and processed in the ER. Now, the key here is that this protein, even though it's misfolded, is often still marginally functional. If it could somehow reach the cell surface, it could potentially transport some chloride ions, maybe lessening the disease severity.
But the RER intervenes before it ever gets a chance to reach the surface.
Precisely. The RER's quality control chaperones, like BP and others in the folding pathway, recognize this slight misfold. The system rigorously deems the protein substandard. So instead of allowing it to be packaged into vesicles and shuttled to the Golgi and then eventually to the plasma membrane, the RER strictly retains this defective CFTR protein. The protein is then targeted and diverted to the ERAD pathway we discussed, retrotranslocated out of the ER and degraded by the proteasome and the cytosol. The tragic result is that functional CFTR expression on the surface of epithelial cells, particularly in the airways and pancreas, is dramatically reduced. And this lack of chloride transport leads directly to the symptoms of cystic fibrosis, like the thick, sticky mucus production.
Yeah.
So the pathology isn't primarily because the gene can't produce a protein at all, but because the cell's own internal quality assurance system prevents the slightly faulty resulting protein from ever being deployed, even if it might have performed its job, albeit imperfectly.
That really puts a chilling perspective on the power and importance of those chaperones and folding checks. Okay, finally, once the products clear all these hurdles, RER quality control, SER modifications, Golgi processing, and sorting those destined for the outside world leave via exocytosis.
Exocytosis is the final step in this whole secretory pathway. The finished macromolecules, packaged neatly in transport vesicles that butt off the TGN, are targeted to move towards and fuse with the plasma membrane. This fusion process expels the vesicle contents directly into the extracellular space. This is the fundamental mechanism for releasing massive amounts of complex molecules. Things like peptide hormones, insulin is a classic example, neurotransmitters at synapses, or specialized components needed for building the extracellular matrix around cells.
Okay, we have completed a really comprehensive tour, I think, of the cell's internal infrastructure, covering manufacturing, specialized chemistry, quality control, and shipping. It's clear that the RER is the manufacturing floor and the initial quality control site, handling the synthesis and essential folding.
And the SER handles the heavy-duty lipid chemistry, the steroid hormone synthesis, those critical detoxification efforts, and the meticulous sequestration and controlled release of calcium ions, acting as the cell's really key internal signaling nexus.
And the Golgi apparatus emerges as this hyper-efficient post office, sequentially modifying products, especially through complex glycosylation, and sorting them with incredible precision based on destination tags, like that M6P system for the lysosome we talked about. It's an infrastructure that demands a seamless, integrated flow of material from start to finish.
And we really must emphasize that this entire complex system, the extensive interconnected mesh of the ER, the ordered stacks of the Golgi cisternae, and all the associated transport vesicles buzzing between them, this is not static machinery just sitting there.
Right, that leads back to our concept of organelle biogenesis you mentioned earlier.
Exactly. In growing and dividing cells, this entire functional system must be accurately duplicated, expanded in size, and then correctly partitioned or apportioned between the two daughter cells following mitosis. It is a constant, ongoing feat of logistical engineering to ensure that the biosynthetic capacity is perfectly renewed, maintained, and divided so that every single daughter cell is immediately viable and equipped to function.
A perfect mechanism for maintaining life, billions upon billions of times over. But I want to leave you, our listener, with one final provocative thought that brings us right back to the topology, the spatial arrangement we discussed at the very beginning. Since the ER lumen is structurally equivalent to the extracellular environment, the ER and Golgi machinery are ultimately responsible for producing the very membranes that define the cell's boundary, with the outside world the plasma membrane itself. Now, consider the incredible complexity required to maintain the precise asymmetry of these membranes at all times, making sure that the correct lipids and the correct protein domains are always facing the cytosol versus facing the lumen, which becomes the outside world. This requires constant active molecular transport, the work of those scrambloses and flippases we touched on, to fight against the natural tendency towards equilibrium and actively maintain the crucial structural and functional difference between the inner and outer membrane leaflets.
It's a fundamental structural constraint that quite literally defines life itself.
Yes.
It just goes to show that in biology, perhaps more than anywhere else, architecture is function. That's probably a good place to leave it for this deep dive.
Absolutely. We'll catch you next time for more insight into the invisible systems that govern our world.